Am I audible?
Yes.
Yeah, you're audible.
So it's my pleasure to introduce Professor Steven Henikoff, a well-known molecular biologist and principal investigator at Fred Hutchinson Cancer Research Center and Howard Hughes Medical Institute. He earned his PhD from Matt Messelson's lab at Howard University and postdoc from Charles Ladd's lab at Washington University. During his postdoc, he got fascinated by a very interesting phenomena, that is position effect variegation in Drosophila. And ever since he has published, and then his interest in, ever since he is pursuing his interest in understanding the mechanism of epigenetic inheritance. And so far now, he has published more than 300 papers in the field of molecular biology and epigenetics. Few of topics are like nucleosome dynamics, nested genes, histone variants, genome evolution, and centromere. Professor Steven's lab is also recognized for developing cutting-edge technology in the field of molecular biology, like recent one, CUT&Tag, CUT&RUN, and his passion towards science has made him move his lab into his house during the pandemic, and during which he established an experiment to profile an epigenome at home. Now, without any further delay, I would like to welcome Professor Steven Henikoff to deliver his talk.
Well, thank you very much for that kind introduction. Okay. Am I coming through okay?
Yeah.
Yeah.
Okay, great.
Yeah.
So let me start out by explaining what I mean by the title, Protein-DNA Interaction Dynamics. Now, all of the active processes that take place on our genome, whether it be replication, transcription, repair, transcription factor binding, they all must take place in the context of nucleosomes, which occupy about 70% of the chromatin landscape. So what we're going to be talking about in large part is the nucleosome core, where you see that there's spiral that the DNA system follows around. And you have the dimers of histones H3 and H4 that form a 4-helix bundle with another dimer of H3 and H4. And above and below, you have dimers of H2A and H2B. Now, the point here is that the DNA is packed so tightly around that it creates an impediment for active processes, for example, replication. So in blue here is the replicative helicase, and you can see the DNA, the double-strand DNA goes through, and it gets extruded.
Steve, I'm going to Steve, I'm going to mute all. You have to unmute yourself and start, please.
Oh, am I muted?
Yeah, Steve, you can unmute yourself now and start. I have unmuted everybody. Just unmute yours and start.
Now, how's that?
Perfect. Yeah, please go ahead. Yeah.
Oh, okay. Yeah, there was some feedback.
Yeah.
Okay, so let me let me continue on here. So what you can see in this video is that the replicative helicase, the DNA goes through that little hole there and then gets extruded into single strands to give you the leading and lagging strand. But of course, if you think about the nucleosome or any protein that's associated with the DNA, it's got to come off. So this is the most dynamic point in the this is the most dynamic time in the cell cycle as far as the chromatin landscape is concerned. Okay. So what we're going to be talking about today is how active processes such as DNA polymerase, or in the case, primarily in the case of transcription, RNA polymerase, how does it deal with the fact that it has to go through a nucleosome, and the nucleosomes block all these active processes, and we've been interested in that interaction. I'm going to start out by talking about the methods that we've applied recently in order to map the chromatin landscape. And then we'll turn to some interesting biological questions. First of all, how does a transcription factor find its site when it's sitting on the surface of a nucleosome? And then what happens to the nucleosome in the process, the unwrapping that has to occur in order for active processes to occur? And then we'll turn to nucleosome assembly, what happens behind the replication fork and at other times in the cell cycle. Okay, so let's ask, how do we map the chromatin landscape? Now, the method that's been around now since the mid-1980s, chromatin immunoprecipitation, has really dominated in the field. And over the past dozen years, this has been coupled to a sequencing readout. Now, with chromatin IP, you begin by cross-linking and then solubilizing the entire cellular components to shatter the DNA into small fragments. Then you add a specific antibody to immunoprecipitate, and then you extract the DNA, add on adapters, amplify the library, and do DNA sequencing. Now, a few years ago, when Pete Skeen was a postdoc in the lab, we came up with a method that we refer to as CUT&RUN, and it's fundamentally different from CHIP in that there is no solubilization. In fact, the cells and the nuclei remain intact throughout the entire process. We permeabilize the cells and then add the antibody, say, to a transcription factor, and then we add a fusion protein between protein A and micrococcal nuclease. Now, the protein A will bind to the antibody, which anchors the nuclease such that when calcium is added to activate the emanase, it will cleave. Now, this was introduced in 2004, this approach by Uli Lemley, and in our version of it, we allow the factor complex to leak out after it gets cleaved on both sides. And then the rest of it, we just take the supernatant and we continue on in the same way as CHIP with the extraction, ligation, and amplify the library. Now, last year, when Hatice Kayo-Okur was a postdoc in the lab, we introduced a variation on CUT&RUN in which instead of using emanase, we use the cut and paste transposase TN5. Now, TN5 is the basis for the Illumina and Nextera system, also the popular TAG-seq method. But in our case, we are anchoring it via the antibody. We're anchoring the TN5 such that when we activate the enzyme with magnesium and it drops its payload of adapters, then we can extract the DNA and amplify the libraries without any ligation required. And this process takes about a day from live cells to sequence-ready purified libraries. And the backgrounds are extremely low. And this is in large part because unlike CHIP, where you are sampling from the entire genome, with CUT&RUN and CUT&Tag, you're targeted just to the region, just to the site that where the antibody is. And this gives us extremely low backgrounds. Now, we've always been trying to improve the protocol. So one thing we decided to get rid of was this DNA extraction step. And instead, we find that by successive additions of SDS and heating it up a little bit to remove the TN5, which is tightly bound to the DNA, to remove it from the DNA. And then we can neutralize that with Triton, so we can do PCR. We can do the whole thing in a single PCR tube, starting from cells, or nuclei in this case, all the way down to sequence-ready amplified libraries. Now, as we were finishing this up, and it was, we just had to optimize a little bit more, the COVID epidemic hit, and I realized that, well, you know, there's nothing here that I couldn't just set up in my laundry room right a few steps from where I'm sitting here. So I threw some of this material into the trunk of my car and brought it home. So this is it. This is all you really need. You need a PCR machine. This is one from the lab. A tube rotator, rubber bands to hold on the tubes. You have a toy centrifuge just to move the material to the bottom. Nothing very dangerous here. A vortex mixer and a couple of magnets and some. This is basically all you need. This is a cooler that we use for picnics. Okay, so this is set up, And then I got started working. I actually just started an experiment this morning. And this is the kind of data that you get. So here is a histone modification, H3K27 trimethylation. It's put on by the PRC2 complex, and these are human K562 cells. And what you see is ChIP-seq data from ENCODE showing that there's a large domain of H3K27 trimethyl. It's a critical mark for polycomb silencing, and I'll be saying a lot more about that later in the talk. And you can see, even though ENCODE requires about 1 million cells, in CUT&Tag, even going down to about 100 cells, we still get very nice signal to noise, but much better than you get with ENCODE. And that's because the backgrounds are extremely low. And we get similar kinds of results for a mark of promoters, H3K4 trimethylation. We can go down to about 100 cells or so, and we can still get peaks, and the signal noise is good. Now, what I'd like to point out here is that some of these were done in the lab, some using this direct-to-PCR protocol, and some were done at home, and some were done by extraction. They all pretty much look the same. So we get high data quality with very little investment in equipment. Because this is so useful, we put this up on our Protocols.io site as a protocol that people can use. Now, Protocols.io is interactive, so you can go to our site. We have sites for CUT&RUN, CUT&Tag, and now for the CUT&Tag Direct is CUT&Tag at home. You can go there, get the latest protocols. You can ask questions. I monitor, Derek Janssens and I monitor, answer questions. We've answered hundreds of questions. They come in every day. But at the same time, in order to give the background to it, we put it up a paper in bioRxiv. So you can, and they are linked to one another. So you can get the protocol, and then you can see what the results look like and get it in the context of a real experiment. And this seems to be caught on. And here's somebody who lives right across the street from Mass General Hospital who set it up on a desk. Looks pretty good. Just show one thing that they did that we hadn't thought of. So in order to get a tube rotator, they have a barbecue rotisserie motor that they stuck a pen into, so that you basically tape the tubes to. So you can do this without a whole lot of equipment. But if you do have equipment, for example, if you just happen to have a robot, you can automate the whole process. So this is work of Derek Janssens, who first automated our cut-and-run method. And this just shows an example where we're doing patient-derived xenografts that get disaggregated, mixed with concanavalin A beads, and then arrayed in 96-well format, put on a robot, and all the reactions were done on the robot. It was taken off to do PCR, put back on the robot to do a cleanup, some QC, then mixed up to 48 or 96 samples and did paired end sequencing. When we price this out, our in-house costs are about $25 a sample, and that turns out to be well within the range in which it's worth thinking of doing it actually as a clinical service. And in fact, we're collaborating with our clinical colleagues in the Fred Hutch Shared Resource in order to profile, to do this cut-and-tag, AutoCUT&Tag on patient samples. And that we're doing that right now. So I think that you're going to see a lot of this in the future. Finally, I should mention that it's quite versatile, cut-and-tag, because the tagmentation takes place inside the nucleus, and there is no release of the DNA. And what that means is that we can dispense in a single well format. So this is using the Takara iCell 8 system. It's a chip that's 72 by 72 nanowells, about a couple of centimeters wide. And this is the image that you see after nanodispensation of cells. So there's some cells where there's some wells with single cells, some with more than one cell. And the reagent for PCR is put only into the wells that have single cells. And then it gets PCR'd in 72 by 72 format to give over 5,000 over 5,000 wells in there. And when we applied this to cut-and-tag, we got very nice data. This shows this shows using this H3K27 trimethylation, this polycomb silencing mark, which silences the HOXB locus in human ES cells, but not in K522 cells. You're looking here at single molecule, single cell tracks, and you can see that we get good coverage even in a single cell. And this shows that we can use it to discriminate cell types. And that's not all, because it turns out that it's adaptable to other readout systems with our collaborators Anu Patel at the Fred Hutch and Jay Shanjuri at the University of Washington. We've applied it for combinatorial indexing and on the 10x, and in both cases we get really nice data. So we think that this is going to be the way to go for single cell profiling. Okay, so those are the tools that we have at hand, and now what we're going to turn to is the first interesting question is, well, how is it that a transcription factor can find its site on the surface of a nucleosome, which it must have to do because the nucleosomes cover 70% of the DNA? Now, there have been a couple of ideas as to how this might occur that have been put forward over the decades. So one is what we might call site exposure, where there's transient unwrapping of the nucleosome to expose the binding site, so just transiently, so the transcription factor can bind. And the other idea is pioneering, where the factor sits on the surface of the nucleosome and uses other parts of the protein in order to displace that nucleosome so that it can remain bound. Now, Mike Mears, postdoc in the lab, realized that he would be able to test these models and other models using CUT&RUN, and here's how he does it. So we differentiate human ES cells to endoderm and then take samples and do CUT&RUN over a 5-day period. Now, what you're looking at here is the results of CUT&RUN for a pluripotency factor, SOX2, and it shows a heat map representation. So what we do here, and I'm going to be showing more heat maps, so bear with me on this. So what we're doing here is we're doing peak calls on the data to find where SOX2 is, and then we are lining up over the midpoints of the peaks, or the summits of the peaks, and we order them by increasing occupancy over the peak. And so you get this nice pattern here. By day 2, you can see that it's gone. Well, it's a pluripotency factor, that's what's expected. But if we look at differentiation factors, such as SOX17 and FOXA1, we see that they kick in over the course of differentiation. And CTCF is an example of a constitutive factor. Now, here's the idea. If a transcription factor is bound to the surface of the nucleosome, then it will not be able to cleave on either side with the MNase that's tethered to it. However, it can cleave around the entire nucleosome, releasing a larger fragment than if the transcription factor is just bound without a nucleosome, which would give a smaller fragment. And we would expect by the pioneering model that at a locus that is pioneered, you would see a shift in size over differentiation from a large to a smaller fragment. But at other loci that are not pioneered, we would just see the small fragments. So here's an example for CTCF. And what we're doing here is plotting the mean fragment length on the y-axis. So larger fragments are above. And what we see is that there's very few sites, loci, that show pioneering by this assay. However, if we look at FOXA2, which is a bona fide pioneer factor, you can see that about 29% of the sites show by this assay show this pioneering activity. In fact, this turns out to be general for pioneer factors. So here what we're looking at is CTCF, and we're using a cutoff of 120 base pairs. Nucleosomes are 147 base pairs, so anything that is larger than 120 we'll consider a nucleosome, and anything below is not, and that would be the transcription factor itself. And so you can see that there's some 8% at CTCF sites, there's some 8% of those sites show pioneering by this assay, going from early to mid to late in differentiation to definitive endoderm. Whereas GATA4, about half of the sites show pioneering by this assay. Now, if we can separate the sites based on whether they're pioneering or not, we can ask what's the difference between those sites? Are the sites, some sites more highly, do they bind the transcription factor more tightly? To test that, we look at the motif that is being, that the factor has evolved for. So for FOXA factor, this is a sequence logo representation of the motif that FOXA likes to bind. And we will make the assumption that this is what FOXA has evolved to bind, and any deviation from it would be lower affinity site. And based on that logic, if we look at the motif scores, what we see is that the pioneering sites in general, for all the factors that we've looked at, show lower affinity for their sites. Now that might seem counterintuitive, so let me just explain how we can rationalize this. A high affinity site would be just an ordinary DNA binding site. But however, if there's a low affinity site on the surface of the nucleosome, other parts of the protein that can recognize the nucleosome non-specifically and bind to it will stabilize its binding. So in the case of FOXA2, it has two winged helix domains. Winged helix domains are found in histone H1, which binds to the entry-exit sites of the nucleosome, binds to the DNA on the surface of the nucleosome non-specifically. So we think that the non-specific binding by other parts of the transcription factor is sufficient to hold it on long enough so that the transcription factor engaged to find its specific site. That way you can get away with lower affinity binding. And we think that that has evolutionary consequences. Lower affinity binding sites are, of course, going to be much more abundant in the genome than high affinity sites. So that increases the search space for finding a new site over evolutionary time. And once such a site is found and does its thing and then goes to fixation in the population, there'll be no further selective pressure for it to go to higher affinity. So these sites will remain low affinity. And so what we imagine is that the factor is going to bounce from one nucleosome to the next, looking for its site, and only sticking on when the site when the site is actually bound specifically, and therefore will stay longer in order for the transcription factor to do its thing. And this could account for the fact that transcription factor binding sites in large genomes like ours seem to be quite degenerate relative to what it would take in order to specifically find a site throughout the genome. And we think it takes advantage of nucleosomes in order to do that. Speaking of which, what happens to the nucleosome in the interaction? And this might be a little bit of a surprise. What we see is using a TAG-seq, which maps accessible DNA, so-called open chromatin, and that's the way it's usually thought of. What we see is that by day one, even though FOXA2 is not expressed at this time, it's only expressed later, by day one, we see that there's a good accessibility site, that the site is already accessible. And it's not accessible in the fact that there's no nucleosome, because we also do cut-and-run for histone modifications, and we see at exactly the same site, there is a nucleosome there. So there's something interesting going on here. We see it's simultaneously both accessible and has a nucleosome, and that doesn't fit with the way DNA accessibility is usually described as being open or closed. It's somewhere in between, and we think this is attributable to the dynamics. That is, that there's nucleosomes, but they are quite unstable, and they allow the transcription factor to bind. And this is from a couple of papers that appeared late last year, which clarify that. In this case, they're doing accessibility mapping, but not with micrococcal nuclease or TN5 or something like that. They're using restriction enzyme. Restriction enzyme will cut its site, specific site, 100% of the time, and will go to completion. So therefore, they can get an absolute occupancy, absolute accessibility measures using this. And in this paper, they show here that if you look at an accessible site using DNase 1 hypersensitivity, same as a TAG-seq, you can see the difference between a hypersensitive site and one that is not is about 20% of the total, somewhere in the not fully accessible and not fully inaccessible. So the dynamics at these sites is actually can explain the accessibility properties. And there was no site in the genome that they could find that actually was completely accessible or inaccessible. You can understand that when I showed you the replication fork going through. At that moment when it's going through, it's completely accessible. And then later, as I'll show you, the nucleosomes will come back in. Okay, and this leads to the next part of the talk where I'd like to tell you about our work looking directly at nucleosome unwrapping dynamics. And this is a story that was motivated in large part by a paper that appeared from David Shor's group several years ago. And this shows a heat map representation, a little different from the heat map that I showed you previously. In this case, what they're doing is for yeast genes, 5,000 yeast genes, they are lining up the first nucleosome downstream transcriptional start site. So what you see here is this is a line through the first nucleosome. And if you go to the second nucleosome, we'll call this the +1 and this the +2 nucleosome, there's a uniform distance between them. However, between the +1 and -1, there's a variable distance, and they've taken advantage of that in order to order by increasing width of the nucleosome depleted region, or as I call it, the NDR. And what they noticed was something interesting. There are these particles here that show up in the wide NDRs, and the particles are about the width of a nucleosome. That's about 150 base pairs. And so they assume that these are nucleosomes, and they showed this interesting property that they're very sensitive to this enzyme micrococcal nuclease, which is used for the mapping. So these are so-called fragile nucleosomes. However, this interpretation ran into some controversy. Another group said, well, we did some CHIP and we didn't see any histones there. They also looked at some data that we had published using a chemical cleavage mapping approach, and we didn't see, and they didn't see any particles in here. So they argued that these are some other kind of particles. We don't know what they are, but they're not nucleosomes. And then Shore came back and said, no, actually, when we do CHIP, we see that there are histones in there. So what's going on here? We had an idea as to what might be responsible for this, and we thought it'd be quite interesting and worth following up. And what we thought was that it's this large complex, a nucleosome remodeler called RSC. Now, RSC is the only essential remodeler in yeast, and its job is to maintain nucleosome depletion at NDRs. And this comes from a recent cryo-EM structure from Patrick Kramer's group, in which you could see that there's this DNA interaction module of the complex that holds the complex onto the NDR. So here's the NDR. And then there's a nucleosome over here, and it reaches around and grabs that nucleosome and uses the energy of ATP to slide it along. Now, the reason that we were thinking of this model was work of Srinivas Ramachandran when he was a postdoc in the lab. He had shown that in yeast, the +1 and -1 nucleosomes are partially, there's a subset of those nucleosomes that's partially unwrapped, and we were able to implicate RSC in the process of the unwrapping. So what we thought was that perhaps these fragile nucleosomes are RSC bound, and because of RSC is this large complex, it's somehow impeding the ability to detect that nucleosome there. So in order to test this directly, Sandapan Brahma, postdoc in the lab, came up with a modification in which we can do chromatin IP after cut and run. It goes like this. In yeast, we use a tag, a flag tag. And so then we use an antibody to the flag tag. And we can get rid of the first antibody by taking the supernatant from chromatin IP, from cut and run, and add excess flag peptide. That will compete away the first antibody, so we can add a second antibody, and then we can do CHIP on that. Okay, so that means that we can look at RSC and we can look at whatever particle is associated with RSC. So here's the result. First of all, RSC cut and run. And what we see is we had previously profiled risk using a native chip approach, and we see basically the same result. Risk is all over the place, all over the yeast genome. However, it does have certain places that it likes to be, such as the +1 and -1 nucleosome. And we get pretty much the same result for CUT&RUN as ChIP-C. But with CUT&RUN, we can take the supernatant and then add antibodies to various histone modifications. So the negative control, IgG, is flat. H2AZ is a histone variant. I'll be talking later about another histone variant, but H2AZ is a histone variant that occupies the +1 and -1 nucleosome and has already been found to be associated with risk. And we see that there's a lot of H2AZ in this risk-containing particle. And we also see some active histone modifications. Now what we're going to do is take the same data I'm showing here, and we're going to line it up in a heat map format similar to what Shore had shown, where we line up over the +1 nucleosome downstream of the transcriptional start site. And what we can see is from our native chip data and for CUT&RUN data, we get the same result, that there's a lot of risk in the wide NDRs, but not in the narrow NDRs. Then with CUT&RUN, we can ask, okay, so what are the histones? Are there histones at all associated with the risk that's in the wide NDRs? And in fact, we don't really see anything. We don't see any difference between the wide and the narrow NDRs when we look at these H2B or the histone variant H2AZ or these active modifications. However, remember what I told you was our model is that the nucleosomes are partially unwrapped. And therefore, here we're looking at all nucleosomes. What if we just to look at the unwrapped nucleosomes, the ones that have less DNA associated with them? So to look at this, we are going to split the data into those with a wide NDR and those with a narrow NDRs, and then we'll average over it and look at the smaller fragments. In this case, less than 120 base pair fragments. And indeed, what we see is for H2AZ, for the wide NDRs, we see that there's a particle in there as predicted. And we also see that for active modifications, and they're rich over H2B. So this confirms that indeed risk is associated with nucleosomes in the wide NDRs, and it's unwrapping, indicating that it's an intermediate in remodeling, and that's why we're seeing the NDRs here. Now, risk is not the only essential factor that's required for making an NDR. Over a decade ago, Hit and Madani at UCSF had shown that there are two general regulatory factors, ABF1 and REB1, that are also essential for making an NDR. And so we looked at ABF1 and REB1 and got a similar result, even more striking. So here we're doing CUT&RUN with ABF1 followed by histone chip and looking at the smaller fragments. And what we see is that right over the NDR, right over the middle, you basically see for the wide NDRs, you see that there's a particle for ABF1 and also for REB1, but we don't see that for the narrow NDRs. So we have a lot going on here. You have this powerful machine that can unwrap nucleosomes. You have the sequence-specific binding factors, basically transcription factors, that are also needed in the process. So how can we sort this out? So what I'd like to do is give you a little bit of background for how we're thinking of it, and this will also introduce you to the last part of the talk. And this comes from work that Srinivas had carried out when he was a postdoc in the lab several years ago, in which he came up with a method for metabolic labeling of chromatin, basically looking at events behind the replication fork. So you remember the video that I showed, everything must come off and then the nucleosomes must come back on. And this basically detects when they come back on behind the fork throughout the genome. And we're lining up over the +1 nucleosome, and now we're doing this in Drosophila. And what you see is that you get the +1 nucleosome, the -1 nucleosome, and the NDR. That's the black line at steady state. However, 10 minutes after the replication fork has passed through, we see that nucleosomes fill in the empty space. And then after an hour, they begin to come back. And although we did this in Drosophila last year, Anya Gross's group showed that the same thing is going on in mammalian cells. So we think that this is typical for animal cells. And in this study, we showed that these nucleosomes will compete with transcription factors, which sort of brings us back to the first part of the talk where I was basically showing that there is this dynamics involving transcription factors. We think that this is a large part of it. And we also found evidence that this is mediated by the BAF complex, which is Drosophilus' only ortholog of the RSC and SWI/SNF complexes in yeast. Now, however, about the same time, another group published a paper where they did pretty much the same approach. They had 5-minute resolution, better than ours. They did this in budding yeast, but they didn't see anything at all like what we saw here. They didn't see anything filling up in the NDR. And we attribute the difference to yeast and animals by the difference between the BAF complex and RSC. And that's because I was pointing out the DNA interaction module that's present that anchors RSC to the NDRs. That interaction module is entirely missing from animal BAF. And we think that makes a lot of sense. What you have is in yeast, the genes are basically, most of the genes are constitutive. It's a single cell organism. And so most of the genes are just always going to be expressed. So they have to be open for business all the time. That's not the case for developing organisms that have multiple cell types, because an NDR at one place is not going to be an NDR, and in one cell type is not going to be an NDR in another cell type. So you wouldn't want RSC there all the time. Now, how does RSC know that there's where to go? Well, it turns out that there are GC-rich motifs and other motifs that it actually recognized. So this is a sequence-specific binding of RSC in the NDR. So what we think it's doing is just sweeping up and back and keeping this clean. And so this is how we think of it dynamically in our model. And that is that behind the replication fork, a new nucleosome will go in. RSC, which will recognize as GC-rich motifs, is on it like a shot. It will then use the energy of ATP to slide that nucleosome along. But in so doing, it will expose the binding site for a general regulatory factor. And we know that Reb1 has a residence time of only 2 seconds. So shortly after it binds, on average, it will come off. But RSC also has the activity in vitro that it can evict the histone core from a nucleosome. So when it does that and the factor comes off, now you're back to where you started. So we think of this as a cycle. Behind the replication fork, a new nucleosome will get put in there. However, RSC will take it out, and then another one will go in throughout the cell cycle. So we have 2 different processes of nucleosome assembly, one behind the replication fork, and then one that continues throughout the cell cycle and maintains this nucleosome depleted region dynamically. And this leads me to the last part of my talk, in which I'm going to show you that there's actually a counterpart to what I just described, and this counterpart is in a histone variant. And that is that there's a histone variant called H3.3. In Drosophila, H3.3 and the canonical H3 differ by 4 amino acid residues. And that is, there's 3 on the core in a cluster, and then there's 1 on the tail. Now, these are extraordinarily well conserved in the sense that the proteins in that H3 and H3.3 in humans is 100% identical and differ by these 4. So these 4 amino acids really stand out there. Just one thing I note so you won't get confused. In humans, there's also an H3.1 and H3.2. H3.2 is identical to what you see here in Drosophila. H3.1 has 1 amino acid difference that we don't think is involved in the process. So what's so interesting about this came almost 20 years ago when Kami Ahmed was a postdoc in the lab. And what he had shown was that if he made GFP constructs to H3 and H3.3 in Drosophila and looked under the microscope, what you see here are these 2 cells that show these different patterns. Now, what's responsible for these patterns? Well, Drosophila has a chromocenter in which all of the heterochromatin, which is late replicating in the cell, will congeal. So you get in Drosophila cells, so you get this chromocenter here. And that tells us it's a late replicating cell, whereas early replicating euchromatin shows this pattern. So we can order these cells, and you can see for both H3 and H3.3, we see the same pattern. However, there's a difference over the rest of the cell cycle in that H3, we don't see any incorporation at all, whereas for H3.3, we see incorporation throughout the chromosomes. And there's one spot there that turns out to be the active ribosomal DNA cistrons. So these are the most highly transcribed regions in the genome, and they really light up. So this tells us that there's two distinct processes. There's one of replication coupled assembly that can be seen for both H3.3 and H3, but H3 does not participate in the other one, which is transcription coupled assembly. Now, Kami confirmed this by making swaps between these residues and showing that single changes from H3 to H3.3 begin to show this pattern, although not the other way around. So these are distinct processes of nucleosome assembly that we can measure using these two variants. Now, fast forward 10 years, and H3 and H3.3 got a lot of attention because they were found to be Oncohistone mutations in pediatric brain tumors. And these are diffuse midline gliomas, or DMGs, and they all have a mutation. Nearly all of them have a single mutation. Lysine 27 becomes methionine, and it can be either H3 or H3.3, although the large majority are H3.3 mutations. Now, if you go through the numbers here, there's 16 histone H3 genes, mostly H3.1, 3.2. However, it only takes a single allele of one of the 16 genes to drive cancer. So it's extraordinarily powerful oncogene. And as you might expect, because I've already introduced you to lysine 27 as being responsible for polycomb silencing, several groups jumped in and showed that indeed that this oncohistone inhibits lysine 27 methylation by the PRC2 complex. Now, it's worth noting that these are unique brain cancers. It can be identified, it can be diagnosed simply as a tumor because it's an unusual place in the brain, and pediatric cancers are unusual. And so it has its own WHO designation. And furthermore, it's exceptionally malignant in that either H3.3 or H3 show very early age of diagnosis, and H3.1 shows an even earlier age than H3.3, so it's even more severe, and more severe than other oncohistone, and at the lower end of the scale of pediatric high-grade gliomas that basically that do not have mutations. And there has been no observation among 1,000 patients of long-term survival. So what makes it so extraordinarily malignant in cancer? And we think it's because of replication coupled assembly. And that's because if either H3.3 or H3, if the nucleosome goes in behind the replication fork, it will pepper the entire genome with an inhibitor of the polycomb complex. And if you inhibit polycomb, you can't silence your genes, so therefore you won't differentiate and remain pluripotent, which is a good description of a cancer cell. So we asked, can replication coupled nucleosome assembly explain the inhibition of lysine 27 methylation in these DMGs? And this is work of Jay Sarthi. He's a pediatric oncology fellow in the lab who really got us going on this project. And what you see is if we show a heat map representation of a high-grade glioma that does not have a histone mutation versus the oncohistone that's either H3.3 or H3.1, we see that relative to the high-grade glioma, we get this low background of labeling for both, but only in H3.3 do we see the active gene pattern. That is, if we line up here in this heat map over the transcriptional start site and order by increasing transcription, we're using histone modification to do the ordering, you can see that we get the active gene pattern for H3.3, but we don't see that for H3.1. It's just distributed throughout. Now, what we're going to do is look, so let me remind you, what we're looking at here is the Oncohistone. Now we're going to look at the methylation. And we do the methylation by calling peaks for the polycomb, polycomb domains. And we're doing k-means clustering, an unsupervised method for these 5 different cell lines. We have a high-grade glioma. We have the H3.3, H3.1 oncohistones, embryonic stem cells, and neural stem cells. And you can see what the high-grade glioma and the neural stem cells have in common is you do see these polycomb silenced domains. However, you don't see that for the H3.3 or H3.1. Globally, they look very much like embryonic stem cells as if they become pluripotent. Now, interestingly, class 4 here shows a difference between H3.3 and H3.1, where H3.3 shows the polycomb domains that you see in normal neural stem cells, but we don't see that for H3.1. Here's an example of that. So there's a polycomb silence domain silencing the GATA3 locus in wild type. And it's also, so we see it in H3.3, but we don't see the methylation. We don't see the methylation H3.1. And this turns out not to be because there's no PRC2, but there is a whole lot of PRC2 there. It must be locally inhibited. So we get global inhibition throughout the genome, and at particular sites, a strong polycomb silencing for H3.1, but not H3.3, we're seeing local inhibition. So let me just explain that with a model, and that is that H3.1 is abundant. It gets put in behind the replication fork. It's made in multiple copies, so there's a lot of it, and it will basically fill up the entire genome. H3.3 is much less abundant, and furthermore, H3.3 is favored for going into active sites. There's almost none of it that will go into polycomb domains. So polycomb domains would be PRC2 that puts the methylation on, would be inhibited by H3.1 at these domains, but hardly at all by H3.3. And this can explain the patterns that we see. So with that, I'm just going to summarize and close. I showed you today that CUT&Tag can be used for sensitive epigenomic mapping, that pioneer transcription factor sites in general have weaker binding motifs, which we think is important for their function. Nucleosome depleted regions are dynamic, as I showed you in budding yeast, and we think that that's quite general for animals as well. And the replication coupled nucleosome deposition can explain the PRC2 inhibition. Okay, the work I talked about involved these people here. I think I mentioned them as I went through. Pete Skeen, who's now at the Allen Institute, and Hadice Kayokur, who's now at the Altius Institute, both in Seattle, really got us going with the CUT&RUN, CUT&Tag. Derek Jansen's automated it. Mike Mears did the pioneer factor story that I told you about. Sandapan Brahma was responsible for the risk study. Srinivas Ramachandran, he has his own lab at University of Colorado, Denver. He really got us going on the dynamics. And Jay Sarthy did the glioma study. And I didn't have a chance to talk about work of other people in the lab, but they're also taking advantage of some of these tools that we developed. We work right here, and these are our funders, and I'll stop there, and I hope there's some time for questions. Thank you.
Yeah, great. So Steve, you can take off the presentation, so then you can see a little better. And those who have questions can. Okay, so Steve, can you hear us?
Yeah, I can hear.
Yeah, so while others may be fixing their mute, unmute business, if I can ask, I find this idea of weak transcription factor sites being facilitated for the binding with the respective transcription factors while the site is on the nucleosome, bit intriguing, because at different sites, the context of these will change a lot, depending upon which part of 146 base pair the site actually lies. So the surface that it will be coming in contact with the histone octamer will be very, very different. It's very difficult to imagine how the transcription factor will be getting facilitated in binding in different contexts of protein surface. On the other hand, only one surface of the one side of the DNA will be able to see, because the other side is hidden by the nucleosome it is associated with. So it appears when it is on the nucleosome, it might be difficult for it to bind, but your data shows it's the other way around. So am I missing something, or?
No, you're not. No, you've got it. You've got it. You're not missing anything. And we've been intrigued by that. We've also had interesting discussions about exactly that result with Ken Zaret, who's sort of the pioneer of pioneer factor binding. And I think we've come to the conclusion that the pioneer factor takes that the function, the role of pioneering is, we might think of as a fail-safe mechanism. That is, the fact that during differentiation, the factor has to bind within a certain amount of time. And if it can't bind at that, if it can't get there in time, there'll be a problem. And the pioneering allows it to do that. And that's because the nucleosomes are not, so the nucleosomes tend to have a rotational phasing of every 10.5 base pairs. So there's going to be a certain favored surface out there, and that's presumably what the pioneer factor is looking for that surface. Now it can be in different parts of the nucleosome where it can get in, but it can't count on in any particular cell that the nucleosome is going to be in the right position. Now think about the nucleosome getting put down, the transcription during replication, when there might be a switch in the transcription factor. That's when we think the window of opportunity is, when the nucleosome is being, during that hour period or so, when the nucleosome is being matured, so to speak. That will give the transcription factor an opportunity to get in, but it might not always have that opportunity to get in, but you got to remember that the nucleosome landscape from one cell to another is going to be different. It's not just that it's at a particular place, but there's a whole lot of places where it's going to be. So the pioneering will allow the factor to get in time for the differentiation process to occur normally, to get in time to actually, so it would basically enforce the timing. So that's why we think of it as a fail-safe mechanism, because sometimes it's going to be available, such as behind the replication fork, but if it's not available, then it will help force it in. So that's the way, that's our model.
Thank you, very interesting. So if somebody has a question, can unmute and ask.
Go
ahead.
I have a question. So Steve, it was a wonderful talk, and I wanted to ask if you have tried mapping at a lot of transcriptomic data and tried to correlate how the differentiation is happening, and what all can you look at the global landscape with respect to RNA levels as well.
We haven't done much with looking at global levels. In a way, we're using this modification, H3K4 dimethylation, which turns out, and trimethylation, they turn out to be actually excellent. It's already known that they are coupled to transcription. For example, it's known that the enzyme that puts them on travels with RNA polymerase II. So because of the methodology that we're using, and we like to compare like with like, we basically do the comparisons to the histone modifications in the studies that we're doing. Nevertheless, we do, when we look at RNA-seq data, which, and the other thing I should mention that we compare it to ProSeq data, which is run-on sequencing, which gets RNA polymerase II itself, but but gets the transcript, and we see correlations there. We don't trust all that much looking at expression from RNA-seq data, because RNA-seq data really comes from the cytoplasm. It's been processed, and there's a lot that's been going on there. So we prefer to look more directly at transcription rather than at the product of transcription.
Okay.
So then I wanted to ask something. Okay, so Steve, if I can ask another one, while others may be still recovering from the fantastic talk that you gave, I'm sure so many ideas that people will be now grouping with this. When we are doing any of these assays, and cells will be in different stages of or differentially kind of different, slightly different versions of epigenome. So how is it that the data looks really good? So which means that the cells really are in different, are not in very different epigenetic or epigenome state, or these things are pretty stable, what we are looking at, and we might be missing out certain subtle things which may or may not be of physiological relevance.
I'm not sure I understood the question. Could you?
I was just thinking that in real life situation, to apply these techniques, let's say in a tissue or something, and not in the cells, can we get this information, relevant information? Because there will be more variations in the in different different populations of cells.
You're asking about accessibility of the data? I didn't quite follow the.
I'm talking about the variations in the different from cell to cell variation in a population when one doing the experiment.
Well, of course, we're getting averages when we do it in bulk, but I did mention that we're doing quite a bit, and with our collaborators, we're doing single cell to get around the problem of cell-to-cell variation. And I didn't have a chance to go through some of the latest data that we're collecting, but CUT&Tag turns out to be really ideal for looking at, because we get so much information out of it, for comparing single cells to one another. And we have several projects going on. One in particular is to look at leukemia, at different MLL rearranged leukemias, and we're seeing very large differences. We're seeing a lot of variation, for example, in patient samples. I mean, it's hugely variable compared to what we would get from cell lines. So, so we're applying it to cancer in order to get at the variability. And I think we're also trying to do this in differentiating system, and other people have done that using a TAG-seq, and we think we should be able to do it using our method.
Yeah, Shagufta, you can unmute and ask.
Yeah, hi Steve, it was a wonderful talk. So basically, I have a question related to the technique, rather, cut-and-run or ATAC-seq. So as everyone knows that somatic cells and germ cells, the chromatin is very differently packaged, and it's more compact, let's say in sperm cells or ovum. So I was rather thinking that if, let's say, someone has to do cut and run, okay, using sperm cells, so the challenges that we would face is that how to basically access those regions in sperm cells wherein the chromatin is so heavily packaged.
I'm trying to think back because we have, so we have.
Have you ever tried with gametic cells or something? Yeah.
I didn't catch that last. You want to know about sperm cells? I do believe that we have users who are doing it on sperm cells. I can't remember whether it was CUT&RUN or CUT&Tag, okay. But if you want to know, go to Protocols.io. It's free, and look for CUT&RUN or CUT&Tag, and there's hundreds of questions that we've been asked, including various things like how do you do it with sperm cells. And I'm trying to remember. I think it was successful, but I don't quite remember what. Of course, in that case, you'd want to know how you actually prepare the
cells.
How you actually prepare the sperm. Yeah. So if you go to the site, if you can find the question, then you can contact the person directly and find out if or get a link to a protocol. But I don't know. We haven't been doing that ourselves.
Okay. Okay. Yeah. Thank you.
Steve, I have a question. This is from Kundan Sen Gupta at ISO Pune. He wants to know how Oncohistone deposition functions in the context of chromatin condensates in the heterochromatin.
Well, actually, it works very. So you're talking about the methods, how they work on heterochromatin?
Yes.
Well, what I showed was our favorite our favorite histone modification is the one for polycomb silencing, and that's facultative heterochromatin, and that's the one that we recommend to users as a positive control because it works so well. We've also been very successful getting good data for H3K27M trimethylation, which is constitutive heterochromatin, and actually examples of that are shown in the bioarchive preprint that I referred to. So yes, there doesn't seem to be a problem, and I think the question is you tend to think of chromatin as being either closed or open, but that's not the case. As I was trying to show you, it's really dynamic. It's also heterochromatin is just more compact, but it's still dynamic enough that the antibodies can get in there. And once the antibodies get in and bind their target, that's really all that's needed. The rest of it, once you anchor the emanase or the TN5, once it's anchored there, then it will go to completion no matter what the local situation is. That's been our experience. So it's a good method for actually getting into heterochromatin. And it works where CHIP is often failed.
Okay.
By the way, we don't cross-link, although you can, that's an option, but you don't cross-link. Cross-linking has been the major reason I think that it's been difficult to get CHIP to work on heterochromatin is because it blocks the epitopes. But if you do things in situ, such that you're in the natural environment, there'll be enough of the dynamics that will allow the antibody to get right in.
Okay.
Hello, Steve. It was a great talk. And my question is related to the transcription factor binding motifs. So how generally it is being decided that they are strong or weak? So either they are sequence conservations which are deciding them, or other mechanisms like small RNAs or something else is guiding the transcription factor to the sites.
I don't think there is any guiding. I think that people not for ordinary transcription factors. There are, of course, there are link RNAs that are involved in, and small RNAs that are involved for some factors. But for sequence-specific DNA binding factors, I think that the model that the model I showed is sufficient together with other factors. So for example, when we compare our GATA4 and FOXA2 data, we can see that there, and also we've done this with SOX2 and OCT4, we can see that there are sites that overlap, that are close to one another and overlap. And we think that they work independently in displacing a nucleosome. And so, so we don't even have to invoke cooperativity. I think that there's enough information there, both by nucleosome binding and by the specificity, in order for diffusion to account for binding. I don't really see any evidence that you need anything more than that. There are cases of guiding by small RNAs, by link RNAs, of course, but not for transcription factor, not for ordinary transcription factors, as far as I know. I mean, zinc finger proteins have get very high specificity because they have lots of binding sites. And we know that they can find their targets right away without any help. So I think the same goes for these for these pioneer factors.
Are there more questions? I don't see any hands raised. So, Saujanya or Santosh, who is managing?
Yeah, if there are no more questions, I would like to thank Steve for accepting our invite to give this talk. It was a very insightful talk. Thank you, Steve.
Thank you.
Thank you. It was wonderful, Steve. Thank you really for accepting it. It was great hearing you after a really long time. And I hope to interact more. Thank you again.
I think there's one question from YouTube.
Okay.
So, Steve, there's a question for you, and they are asking if the differences between animals and yeast nucleosome occupancy, do you see any difference? And what's the idea in plants?
Plants, as far as we know, would be very much like animals for making a nucleosome depleted region, and for the events that occur behind a replication fork. But I don't know I can't think of any data set that's actually shown that. But I think the one thing that makes me think that they'll be the same is that they have the same BAF complex, the same kind of BAF complex, without this DNA interaction module that's very special for fungi and yeast. So I do think that they'll be much like animals in that regard. And it makes a lot of sense to me because they have different cell types, of course, and so they can't have something that's going to sit in all the NDRs and keep this machine doing this cleanup cycle.
All right. Any more? Okay, so I think there are no more questions. So again, thanks Steve a lot for accepting our request and talking to us. Wonderful, really refreshing and nice hearing so many nice ideas and creative ideas.
Okay, thank you, Rakesh.
Okay, guys, so we'll close this. I'll stop the recording.